Bismillahirrahmanirrahim. Hello and welcome back, friends. Welcome back to another video lecture by Microbiologist. I hope all of you are well and fine. Today I'm going to explain another lecture that is related to the staining techniques, and it is about the Albertt staining. If you guys remember, we already talked different stainings, like we have talked Gram stain, Gemsa stain, Leishman stain. Okay, if you want to watch all these staining techniques, so there will be a playlist into my YouTube channel, and the playlist name is staining. So when you click on it, so you will find all the staining techniques. Today I'm going to explain Albertt staining. So what is Albertt staining? What is the principle? What type of the staining are used in the Albertt staining? What is the test procedure? Okay, so my name is Muhammad Kamil Khan, and you guys watching my channel Kami Microbiologist. Let's start the video. Okay, so dear viewers, first of all, let's see that what is actually Albertt stain. Let me explain the introduction. Albertt stain, as the name indicates, Albertt stain. So it is actually the staining technique which was discovered by a scientist named Albertt in 1920. Okay, this type of the staining technique is especially used to find out the chronium bacteria, especially that can cause diphtheria. Diphtheria is actually the upper respiratory tract infection, or we can say it is the infection of nasopharyngeal area, which can be the nasal area and the throat area. Okay. Now, actually, the Albertt collect the specimen, or they collect the specimen from the nasal area and from the throat area, and then they make a smear. And then they stain this smear with Albertt staining solution A and solution B, and then they observe this slide under the microscope. Under the microscope, they found, as you can see on your computer screen, they found these types of the bacterial structures, and they call these bacterial structure chronium bacteria. Now, this Albertt staining is actually stain one of the special type of the granules that is present into the chronium bacteria, and it is the metachromatic granules. Okay, this metachromatic granule is also called voltaic granules, and it is actually present in the cytoplasm area of the chronium bacteria. It is called metachromatic granule because this metachromatic granules have special types of the property which we call it metachromasia. Means if let's suppose their color is blue, so by staining it with Albertt stain, so then its color will be red or pinkish. Okay, so this is actually that's why it is. Next, this metachromatic granule is also called bipolar granule. Why? Because when it is stained, so as it is a rod in shape, so at both sides of this bacteria there will be a round shape, which we call that's why we call it bipolar granules as well. Okay, this is a little bit introduction related to the metachromatic granules. That is really important to identify the chronium bacteria. Now this let me a little bit explain that what is chronium bacteria. So it is actually a bacteria that is rod shape, which we call it bacilli. That is the non-spore forming bacteria, and it is the gram positive and non-motile, means it cannot be moved. Moreover, it contains granules which we call that metachromatic granule, which I already explained. And this metachromatic granule is actually contain a polyphosphate, and it also contains enzymes. Okay, this metachromatic granules can store the food as well as it can store the energy, and it is really important for the chronium bacteria. And due to the presence of this metachromatic granules, the Albertt staining is really it can be this Albertt staining can be catched by this metachromatic granules, and due to this way we can easily visualize this bacteria under the microscope due to the presence of this metachromatic granules. Now, this chronium bacteria can cause a type of infection which we call a diphtheria, and this infection is especially the upper respiratory tract infection, or we can say it is the nasopharyngeal infection, means the nasal area and the throat area infection. Actually, this type of bacteria can also be grow on selective media, on a special selective media, like we have Loflier agar, molar met agar, tensidyl tolerite agar. These are the special types of the selective media which are specially used for the Chronium bacterium diphtheria. If let's suppose you do not have these types of the media, so then you can easily, so then you will just collect the sample or specimen from this nose or throat area where there is the problem, and then you will make a smear on the glass slide and you will stain it, and then you will easily observe the bacteria under the microscope. This is a little bit introduction related to the Albertt stain. Now let's see that what type of the staining we are using in this Albertt stain. There are two types of the staining that are used in the Albertt staining, which we call it Albertt staining solution A and Albertt staining solution B. Albertt staining solution A contain toluidine blue, which is 0.15 gram, malachite green is 0.20 gram, glacial acetic acid 1 ml, alcohol which is 95, we can also use 95% ethanol, that is the 2 ml. So you will mix all these ingredients in distilled water, and then you will wait for 1 day. After 1 day you will filter it, so then you will get Albertt staining solution A. Next we have Albertt staining solution B. So it is it contains the iodine, it contains potassium iodide. Iodine 2 gram, potassium iodide is 3 gram. You will mix it into the distilled water, and then you will easily get the Albertt staining solution B. This is just that how you make this staining solution A and staining solution B. Now let's see the principle of this Albertt staining. So the principle is that this staining contains two basic dye. One we call it toluidine blue and malachite green. These two dyes have acidic property. Why? Because it contains the acetic acid. When the Albertt stain is applied, so what will happen? The toluidine blue will react with the metachromatic granules, and malachite green will react with the cytoplasm. So after that, due to the metachromatic property, what will happen? The bacteria means this metachromatic granule is, if it is blue, so it will be appear into red pinkish color. Okay, so this is the property, this is actually the principle of this Albertt staining. Now let's see the test requirement, that what we need when we are performing this technique. So we need a clean slide, we need wire loop, we need bacterial culture, we need personal protective equipments, we need Albertt solution A, Albertt solution B, we need tissue paper, all immersion, and microscope as well. So these are the most important material that we need in this technique. Now let's see the procedure. So the procedure is very simple. First of all, you have to take a clean glass slide and you will label it. Then you will take if let's suppose you have the sample, if let's suppose you have the specimen on a swab, or if let's suppose you have the specimen, you have the bacterial colony on an agar plate. So what will you do? You will just put a water drop, distilled water, 1 drop of the distilled water, and you will take the bacterial colony and you will mix it into the water drop onto the slide, and you will make the smear. You will heat fix it, so the smear will be easily made. What will you do? Then you will take the slide onto the staining rod or the staining rack, and then you will stain it with the first Albertt staining solution A. You will stain it with Albertt solution A for 7 minutes. You will wait for 7 minutes. After 7 minutes, you will discard this solution. Remember, you will not wash this solution with tap water. Okay, you just discard it. After that, you will add Albertt solution B and wait for 2 minutes. After that, then you will wash it. Okay, in the first step you will not wash it, and in the second staining then you will wash it with slowly running tap water. Now once you wash it, so then you will dry it with the filter paper. You will just wrap this slide with the filter paper, and then you will wait it. Once it is dry, so then what will you do? You will place it on the stage of the microscope and will add the oil immersion. Then you will observe it under the microscope at 40 or 100 x power, 100x oil immersion power. So this is under the microscope, so then you will find the bacterial shape like this, as you can see on your computer screen. So as you can see that the bacteria will be appear round shaped blue-black dots at the both side, and you will see the L-shaped, V-shaped green bacilli. Moreover, this means this bacilli will be look like a Chinese letter. Okay, so this is how you will found the bacteria like in this shape. This is all about the Albert staining techniques. Thank you so much, dear viewers, for watching this video. I hope you understand, but if still you have question, you can write your question into comment. I will try my best to give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and subscribe the channel to get more interesting videos like that. Thank you so much for